Hi, everybody. Welcome to our 2021 SoftBones virtual patient meeting. I'm Deborah Fowler, and I'm the founder and president of SoftBones. And for those of you who don't know me, I'm also mom to Cannon. He was diagnosed with HPP at just 18 months old, and just a few weeks ago, he turned 16. So we've had quite a journey with HPP over the past 13 or so years. I want to start by thanking you all for your generosity and continued support over the past year, which has been extremely difficult for nonprofits, including SoftBones. Your words of encouragement, your financial support, your generosity, the handwritten notes that you've sent to us to encourage us along the way just have really kept all of us going. So thank you so much. On a positive note, we are extremely excited to announce that we have already secured a location for our 2022 patient meeting, which, fingers crossed, will be in person. So drum roll, please. The location will be in Orlando, Florida. So Disney, SeaWorld, Universal Studios, here we come. All of the details for the 2022 Orlando meeting will be found on the HPP and Me community portion of our website found at softbones.org. So stay tuned for more details and information. We're really excited about the thought of getting back together. If you've been to a patient meeting, you know how magical those moments are to connect with others who have HPP. And if you haven't been, I can't even put into words what happens when you see a child connect with another child, or a parent or an adult connect with another adult who has HPP. It's so meaningful, and the bonds that are created just really empower us and help us to live better lives with HPP. So back to the meeting, there's a lot to explore on this platform. There's booths, there's lounges. Be sure to visit the photo booth and take a picture so we can see your smiling faces. Of course, be sure to stop by the SoftBones booth. Introduce yourself to me, Denise, Sue. We would love to say hi to you and introduce ourselves if you haven't met us already. And a special thank you to our generous sponsors who make this meeting possible: Alexion, Panther Rare, and Charles River. Thank you so much for your support. So enjoy the meeting, everybody. And again, thank you so much for being here and for your support. Hi again, everybody. Quick wardrobe change and makeover. Welcome back, and we're so glad you were able to join us today. Before we get started, I'd like to introduce Marnie Edelhart, and she's going to review some of the key functionality of the platform. Marnie?
Thank you so much, Deb. And thank you, everyone, for joining us here today. This is such a special event, and we here at Momentum are really grateful to have this opportunity to work with the SoftBones team to bring this event to life in the virtual space. Before we begin, I want to make sure you have all of the information you need to make the most of this interactive event platform. The agenda is really your starting point for joining all of the program sessions. Sessions only become available to view at their specific start time, at which point you can simply click the Join Session button, and when the session ends, click Leave Session and return to the agenda to join the next one. During each session, you'll have the option to chat with fellow attendees and ask questions of the speakers. These options will appear to the right hand of the presentation screen when you're in a session. I see we've already got people sharing where they're joining from now, so please keep the chat and the questions coming. In between sessions, we encourage you to take advantage of other platform features. In order to interact with each other, all of these features are accessible from reception. The exhibit hall is where you can visit the partner booths to learn more about SoftBones and more about the partners who make this event possible. The event feed is a great space to share photos from past events, share your thoughts about the content. You can post polls, you can post reactions to the sessions, you can post photos of your work setup. You can really have a lot of fun in the event feed. And if you want to get face-to-face chatting with your fellow attendees, the lounges are the space to do it. The SoftBones team has made sure there's a bunch of different lounges available for you where you can get on camera and really have that roundtable chatting feel you probably are missing from live conferences. I know I am. Finally, if you have any issues at all during the event, we really encourage you to visit our chatbot. The chatbot has a true, real, live human behind it. So if you ask questions, it's one of our team answering back, and they will answer immediately and make sure you have all the information you need to enjoy the show. Last, I want to second Deb's mention of the photo booth. There's a photo of me in there. There's photos of all of the people behind the event. It's a lot of fun, and we really want you to get in there and take a picture and connect with everyone, show them your fun side. With that, let me welcome back Deb to officially kick off the event.
Great. Thanks so much, Marnie. I want to thank everybody who joined us last night for our vision board exercise. It was so much fun. It was great to hear about everyone's goals and visions and to connect with everyone. And I want to encourage those of you who were there to share your vision boards on the event feed that Marnie just talked about. That'll be a great way to learn about each other and to continue to engage during the meeting. Without further delay, I'd like to introduce our keynote speaker, Dr. Eric Rush. Dr. Rush is a clinical geneticist at Children's Mercy Hospital and the University of Kansas Medical Center. He's an associate professor of pediatrics at the University of Missouri-Kansas City School of Medicine and a clinical associate professor of internal medicine at the University of Kansas School of Medicine. He is board certified in pediatrics, internal medicine, and clinical genetics. He's the co-director of the Children's Mercy Skeletal Dysplasia Clinic and the medical director of the Children's Mercy Office of Faculty Development. Among his interests are osteogenesis imperfecta, hypophosphatasia, X-linked hypophosphatemia, cancer genetics, cardiovascular genetics, dysmorphology, personalized medicine, and genetics of common diseases. His research interests include extraskeletal manifestations of osteogenesis imperfecta, best practices treatment in hypophosphatasia, skeletal pathology in GFPD, and molecular characterization of rare syndromes. So without further ado, Dr. Rush, welcome.
Thank you. I really appreciate that kind introduction, and it's really good to be here and joining everybody this morning. I wish we were all joining in person, and I'm certainly looking forward to joining everybody next year in sunny Orlando, especially since we're having a pretty rainy morning here in Kansas City. So without further ado, I want to talk to everybody this morning about really just about kind of an overview of what we know about HPP and what we think we know about HPP now. And I really look forward to people's comments on this, and because I think that there'll be some questions, and I think that's a really good thing. And I'm going to separate this into three separate categories. So one is common signs and symptoms of HPP, and in many cases, these are things that we've known about for a number of years, what we will call conventional wisdoms, and some of them are probably true. I also want to talk about some recent learnings about HPP. This, for me, as a clinician and as a researcher, this has been a really exciting time in HPP because I feel like we've learned a lot about this condition, learned a lot about how it behaves, learned a lot about the underlying genetics, and we've learned a lot of things that really kind of challenge some of our conventional wisdoms about this as a purely bone condition. And then lastly, I want to talk about an introduction to genetics and genomics. In many ways, this will be a primer to genetics and genomics. Sort of in a lot of ways, it's kind of a condensed version of some of the first lectures that I give to the undergraduate medical students. And we'll talk a little bit about how we got to where we are today, and then also a little bit about how we understand genetic testing results, because as many of you have found out, it's not that cut and dried. It can be really challenging to understand those results. So we're going to start off with some common signs and symptoms of HPP. And I'm going to use the nosology that we've used for a number of years. I think a lot of you are familiar with this classification of HPP, which is very much based on age of onset. And age of onset is being used as a surrogate for severity, and that's not a terrible way to do it. It's not perfect. There's some arbitrariness to these cutoffs, but I think it gives us at least a sense for what the likely findings and likely severity that we're going to see based on when a person presents with disease. This could be anywhere between those patients who present with perinatal HPP, or what I prefer to call it, perinatal HPP with respiratory failure. And those are our patients that will present in utero and at birth. Then we can contrast this with benign perinatal, or what I prefer to call perinatal HPP without respiratory failure, who will also present in utero and will have disease of varying severities that's present at birth. We can contrast this with infantile HPP, which is still earlier onset, but may not have some signs or symptoms in the perinatal period. And then the largest categories we think are going to be those childhood or juvenile HPP patients, and then people with adult HPP that present later on. And as you all know, odontohypophosphatasia is a little bit different in and of itself. So when we think about perinatal HPP, or again, what I like to call perinatal HPP with respiratory failure, these are our patients who will present very early with severe disease, very poor mineralization. Many cases, there's fractures that are present, and the number of fractures can really vary in these patients. Once they're born, we can see hypercalciuria, so a lot extra calcium in the urine. We can see calcium deposits in the kidneys. These are the patients that are at significant risk for B6 responsive seizures. These are also patients that as my kind of description describe, these are patients that present with pulmonary hypoplasia. Their lungs are very small and they have a lot of difficulty with breathing. And so this can be, in many cases, one of the things that really gets them into trouble. And furthermore, these are patients with lots of problems with muscle tone. These are patients that also have relatively poor growth velocity. And so they just don't grow very quickly. And so they can end up with some significant short stature. The patients with infantile HPP are a little bit different. These are patients that will present, again, with very poor bone mineralization. These are also patients in which we'll see craniosynostosis. Again, craniosynostosis, is, as many of you know, is when the bones in the skull fuse too soon. So all of us are born with a lot of bones that are kind of, in many cases, sort of floating around, if you will, and so they're not fused, and eventually they do. Eventually, all of us kind of lose our soft spot, and we have a fully formed skull. But that happens on a particular time frame, and when it happens early, and when it happens in a way that is asymmetric, that can get people into problems. And if lots of sutures are involved, then you can really get into problems with the way that the brain needs to grow. And if one of them is involved, a lot of times the brain will find a way to grow okay, but the shape of the skull can be very unusual, and that can cause other problems. These are also patients that can have fractures. These can have the same buildup of calcium in the kidneys. These patients can also have seizures and small lungs, and these patients may also have low muscle tone. They may not grow very well, and they may have failure to thrive, difficulty with growth and gaining weight. Those are the patients that will present with the most severe forms of HPP. Now, again, like everything else, there's a spectrum, and the spectrum will kind of flow into what we'll call childhood or juvenile HPP. These are many of the patients that will present classically with rickets, although not everybody has rickets, as we found out. These are patients that can also have fractures, although as we found out, it seems as though patients with childhood HPP have fewer fractures, perhaps, than people with other brittle bone disorders, even though they may have pretty significant disease. These are patients that have been classically described with premature tooth loss. They can have other dental concerns. The teeth can be very fragile. These patients can still have too much calcium in the urine, and this can cause problems of calcium buildup within the kidneys, and that's obviously not going to be good for their long-term kidney health. Some patients can have short stature. Again, I see a really wide range of growth. We see patients that are a little on the short side. We see patients that are a little on the tall side, and we see everybody in between. These are patients that may also have what we'll call proximal myopathy, which is sort of a fancy way of saying something seems to be up with the muscles. It seems to be in the muscles that are closer to the center of the body, shoulders, trunk, hips, places like that, as opposed to what we'll call distal, or this can be things like hands, feet, arms, legs. And they can have lower muscle tone and delayed motor milestones. And these patients can also have significant joint pain. Adult HPP is also a very different entity, even though it's recognizably the same condition. We start seeing, at least in my opinion, we start seeing a lot more struggles with fractures and pseudofractures in the adult population that we don't seem to see as much in the children. Fractures may also heal very poorly. Many of you who are listening this morning may have experienced those fractures or pseudofractures, and you may have had struggles with those fractures healing poorly. These are also patients with adult HPP that may have premature tooth loss, or may have loose teeth, may have things like periodontal disease. Still, too much calcium in the urine can be a problem there. Chronic pain and muscle weakness is a common complaint. And again, joint pain and buildup of calcium and phosphorus within the joints, or something we'll call calcium pyrophosphate disease. And this can be a direct result of some of the issues that are related to ALP enzyme not functioning very well. And we can talk a little bit about ALP a little bit later. And then a word about odontohypophosphatasia, and actually kind of a caveat about odontohypophosphatasia, or at least how I think about it. So defined HPP that is limited to the teeth. And so I feel like I have seen patients where their disease is limited to the teeth. And so I'm kind of operating on the hypothesis that this is a real phenomenon. But I think we also have to be very careful that this diagnosis is given in the context of a very thorough evaluation, trying to uncover other signs and symptoms of a different kind of HPP. And that's one of my worries, is that sometimes patients with presumed odontohypophosphatasia are actually really struggling, and I don't want to see that. The other thing that we have to do is be willing to reevaluate. So you may look at a patient with only dental involvement and say, actually, they look great other than the teeth. And that's wonderful. If you really do that thorough evaluation and they look great, I'm happy for that. But I think we need to be willing to go back and say, hey, let's reevaluate. Let's see what's going on in a couple of years. And maybe that patient's still doing great, and that's wonderful. But maybe that patient's starting to struggle, and we need to rethink our diagnosis of odontohypophosphatasia. So I always encourage colleagues to just be cautious about how we use this term when we're evaluating patients, and also understanding that we're diagnosing these patients at a point in time, and this may not be something that they experience throughout their entire lifespan. So what have we learned lately? So these are kind of some of our conventional wisdoms of HPP. Many of them are probably true, but I think we've learned some additional things that are very interesting, and some of them we're going to talk about a little later in this in this session, not by me, but by some of my colleagues who we're fortunate enough to have today. So aside from our B6 dependent seizures, we have learned that there is concern over different neurological consequences of HPP. And there's a lot of these. And as you can see here, this is not necessarily even an exhaustive list, but this is a list that was reported by some colleagues of mine who are doing this a very fine research at Vanderbilt. So these are patients who will describe muscle pain and weakness that we kind of knew, but we're also seeing things like fatigue that's extremely common, headache is extremely common, sleep disturbance is common, even things that would seem to be completely unrelated like vertigo. Why would patients with HPP have vertigo? Well, we don't exactly know. But 42% of people describing this is very interesting. And then we've also discovered that there seems to be an increased rate of things like depression and anxiety in people with HPP. These are relatively common in the general population, but 39% and 35%, at least visually, that strikes me as being a bit more common than what we would see in the general population. And we'll be talking later about neuropathy, which I know is a topic of great interest to many of you. And then even things like hearing loss, memory loss, tinnitus are things that people with HPP may be concerned about. A different discussion that, again, we are going to hear more about later in the morning discusses specific findings with children with HPP. And these are a heterogeneous group of 30 children between the ages of 3 and 16 that were evaluated with a set of questionnaires about a lot of things like sleep pain, emotional behavioral function, adaptive function, and quality of life. What was found is over half of these children were poor sleepers. About a third experienced a higher than average level of pain interference, and there was a significant prevalence of irritability, depressed mood in this population, and half of the children met criteria for ADHD. These are in my opinion, very, very striking findings, and I think, and I'm very interested certainly to hear more. But I think this is a topic of a lot of interest to us. And we're also learning a little more about the muscular complications of HPP, and in the past this is not considered a true myopathy. So a myopathy kind of implies that not only are there symptoms from a patient from their muscles, it also implies that when we look at the muscles objectively with things like a muscle biopsy or a test like that there's something that we can observe there. And in the past, we haven't really consistently seen those things, not really associated with a specific change. But we've come up with at least a hypothesis for why a person with HPP might experience some of these things, and this can include the muscle tone issues, weakness, and pain, which have been attributed to things like pyrophosphate and phosphate metabolism. And I think that's, at least from a hypothetical standpoint, a reasonable and plausible mechanism. But I think there's a lot we still don't know about why these things happen, and that's definitely a topic of much interest. So what have people been describing in terms of muscular complication? Well, this could include the weakness that I've described in the past, and it seems like it's predominantly in kind of the central part of the body, and what I called before proximal, and especially seemingly in the hips. It's going to include things like low muscle tone, which we observe in a lot of people with HPP, particularly our younger friends with HPP. Pain. There's a kind of an HPP gait that I've certainly learned to appreciate. I think many of my colleagues have as well. That kind of speaks to some of the weakness in the hips. Some poor exercise tolerance, and also there's what we call normal CK levels in the blood. What does this actually mean? Why is this important? Some of our of known muscular disorders, and we can even include things like muscular dystrophies. There's a tendency for the muscles to exhibit ongoing damage, and that damage can be shown by elevation of a certain enzyme called CPK, or creatine kinase, in the blood. But with HPP, it doesn't seem like there's really a trend for that to happen, and so it's not known whether this is a result of ongoing damage to the muscles or whether it's something very specific about how the muscles function. So that's very interesting. And certain other tests can show muscle changes. Again, maybe not super consistently, but at least some of them have shown that. So how have we responded to this? Well, there's been a lot of interest in a large animal model for HPP. As many of you know, some of the HPP research has been conducted on a mouse model. Well, we are very used to doing research on mice and other rodents, but mice are not really small humans. There's really a lot of differences in the way mouse bones work and mouse bone physiology work that make mice not maybe the optimal way to study this. And so there's been great interest in creating a large animal model for HPP. And so what a group down at Texas A&M did is they created a sheep model for HPP, which is very, very interesting. And so in this case, these are animals that were created and you can see here on the right hand banner, you can see a wild type sheep. You can see what's called a homozygous, which means both copies of their Alpl gene are not working correctly. You can see a heterozygous, which means only one of the copies of Alpl is not working correctly, and you can see what's called a compound heterozygous. What compound heterozygous means, and we'll get into this when we talk about the primer, it means that you have both of your copies of Alpl that aren't working. There's a change on them, but it's a different change on each copy. And so sometimes we see a little more variability in these cases. And so it's an important way to look at this. So why do we create the model? It's because we want to look at those muscle and dental changes that we don't necessarily see quite as readily in the mouse model, because sheep, as you might imagine, are a little more similar to humans than mice are. And what was found is this model really does do a pretty good job of predicting the clinical phenotype of childhood HPP. I've seen what some of the researchers for this model, I've seen them kind of demonstrate, even showing things like videos of the animals as they're interacting with them, and they really do much more closely resemble a human with HPP than I think the mouse model does. So it's pretty exciting, and it allows us a little bit of a window to be able to see what we think HPP is like, and just a little more about what the natural history is likely to be. And I think that's a really important aspect of this, because we can't really understand as much about humans until we understand a little bit more about what's going on in a model organism, and we just have such more ability to sort of tinker with this with this model organism that really wouldn't be ways that would be appropriate or ethical to do for humans. And if we look at the muscles in these sheep are different than humans. And they showed, because these sheep showed signs of muscle weakness, they showed signs of gait. And when you look at these little sheep walk, it really does remind me of how a lot of our young children who have HPP walk as well. Obviously, 2 legs and 4 legs, a little bit of a different gait, but it at least gives you an idea. The structure of the muscle is also a little different, and in particular, one of the things that's interesting to me is there were changes in a little part of the cell called the mitochondria. Some of you may have heard of mitochondria before, and I think of things really very simply. And in this case, the mitochondria are little power plants within the cells, and they make all the energy. And these mitochondria are a little bit different compared to normal sheep. Well, what might that do? Well, if you have mitochondria that aren't really working quite correctly, you might be concerned that there is suboptimal energy production within the cell, and maybe that makes those muscle cells not work quite as efficiently as people without HPP. Again, this is really hypothetical. We're really very early in the exploration of this HPP sheep model, and we're pretty early even in the discussion of how are muscles altered in HPP. So I think this is going to be an area of great interest to me over the next few years. I'll really be interested to see this. But that's how we're looking at the HPP sheep model. And how does this bring us to what we think about the care of patients with HPP? And I think what we've discovered, certainly what I've learned over the last few years, is it really takes a village to take care of a person with HPP, because we've learned that this is not just a bone condition or not just a muscle condition or not just an anything. This is a multi-systemic inborn error of metabolism. And I've encouraged colleagues, and those of you who have talked to me in person will probably have heard me say this, that I really consider this to be a multi-systemic inborn of metabolism, like many inborn errors of metabolism that I see as a clinical geneticist. And those inborn errors of metabolism that are multi-systemic require a lot of expertise to really take optimal care of. And I think that it starts with a kind of a focal person who's sort of quarterbacking the team. In most cases, that's either a geneticist or an endocrinologist. That's not always necessarily the case. In some cases, I have some wonderful colleagues that are nephrologists, that are orthopedic surgeons, that may be general internists or general pediatricians that take excellent care of people with HPP and are really doing a great job as their quarterback. But just in most cases, we are seeing that focal point is a person who's a geneticist or endocrinologist. But none of us can function without input from our colleagues, and this requires input from, say, pulmonology, especially in our patients with more severe disease, ophthalmology, because we know that there can be some minor eye findings in patients with HPP, audiology, nephrology, neurology or neurosurgery, again, particularly at our patients who are more severely affected. Orthopedic specialists are often important. Even though we don't necessarily see that patients with HPP are requiring as many orthopedic interventions, surgical interventions, than people with other bone conditions, we still see the importance of them following up with somebody who has a really deep understanding of the way bones work and the way bones work together. Rehabilitation is also very important for patients with HPP, and so I'm a heavy user of physical therapy and occupational therapy services. And in many of our patients, right after they are born, they may have an extended stay in the NICU, and so our neonatology colleagues are really important. And I would be remiss if I didn't mention genetic counseling. So I'm a geneticist, and so I work in tandem with genetic counselors, and I absolutely could not function if I didn't have the support and expertise of my genetic counseling colleagues. They're really important to the process, and they're really important for patients in understanding more about their disease, and in many cases, understanding more about the genetic testing. Because in a lot of cases, it's not necessarily the geneticist or the endocrinologist who's explaining the genetic testing results to the patient and their family. It may be the genetic counselor, and I think that's a very important and should not be understated. And then our dental colleagues. Our dental colleagues are important for the management of the patients to ensure dental health. But I found that our dental colleagues are really important to diagnosis and finding those patients, particularly those patients who are presenting with childhood HPP, in whom the early tooth loss may be their first sign or symptom of HPP. And if you have a really smart pediatric dentist that recognizes that, you can be really ahead of any of the major issues. And so that's a really good thing. And so our dental colleagues are extremely important. So I'm going to spend the rest of this talk about the next 20 minutes discussing the genetics and genomics of humans to start out with, and then we're going to talk a little bit about HPP. And I'm going to try to integrate some of the questions I've gotten over the years from patients about the genetics of HPP specifically, and about inheritance pattern of HPP, because these are actually really complex topics. But we're really going to start off, again, we're starting off with kind of a condensed version of the first 2 lectures that I give to undergraduate medical students, just so they understand a little bit more deeply about what genetics is. And really what genetics is, the study of 2 things, heredity and variation. And so so heredity means the tendency of offspring to resemble their parents. And we all know, we can look around at our family reunions and see heredity. But we can also see variation, which is the tendency of offspring to vary to some degree from their parents. And that's important. And I used to have, and I need to find it, a picture of my 3 kids when they were babies, and then a picture of them now. And when they're babies, they look almost exactly alike. And as you see them now, a little bit older, they nobody's gonna mistake them, that they're gonna know their siblings, but they do look different now. And so there's that almost tension between heredity and variation that it's important to think about when we're thinking about genetics. And it's interesting that the term genetics is not very old. It's only about 100 years old. And it was coined to describe the study of the heredity portion of this. And it's from, like many other things in medicine, it's from a Greek word called geno, which means to give birth. Kind of makes sense. And some terminology is important here too. So when we're speaking about these things, we'll talk a lot about genetic terms, and we throw around a lot of terminology when we're talking about genetics. And sometimes that's unfortunate because it serves to obfuscate, not to clarify. But we talk about things like genotype. And what genotype is just the makeup or composition of a person, whereas phenotype is actually what you see. It's what a person exhibits. Those can be different. And that's actually very important for HPP, because we may see people with the same genotype, the same change in the Alpl gene, but a different phenotype. One person may have HPP, one person may not. And we also talk about things like mutation. And I don't really care for the term mutation. That's kind of fallen out of favor with us. But for the sake of our discussion this morning, we can say that a mutation is just some alteration in the genetic code for what is expected. And what is expected may be what we refer to as the reference sequence. And reference sequences, there's a whole lot of issues with that, because that assumes that one person is more normal than another person, and that may not necessarily be the case. And I don't really like the term normal when it comes to genetics, because we're all a little bit different from one another, and that's okay. But when we're talking about mutation, we're not necessarily judging that. We're not necessarily saying that a change is bad or is causing problems or is pathogenic. So what we use is the term variant. And so you'll hear me talk a lot and use the term variant. And that just means any change. It makes no judgment as to how the change is likely to behave. It just says it's there. And in fact, most changes or most variants are what we call benign, which means they're not responsible for any sort of disease. Some of them are pathogenic and are responsible for disease, and some of them are variants of uncertain significance. And so we don't really know, and there'll be more on this later. We'll talk about that. And one caveat is also genetics versus genomics. I see this used interchangeably in the lay press. Kind of popular media will use the terms interchangeably, but this is not really correct. So genetic testing is what most of you had when you had testing for HPP, which means you're testing the Alpl gene. You knew that's what you wanted. You're looking for known areas of function. That's a genetic test, not a genomic test. But if for some of you that have had things like whole exome or whole genome sequencing, where we're looking at everything, we kind of consider that genomic sequencing. So you're looking at large amounts of genetic information across the entirety of the genetic material. And you may be looking at things that have known function, but you also may be looking at things that don't have known function. And so you may not be exactly sure. You're kind of casting them that a little more broadly. So that's kind of the basic difference between genomic and genetic testing. But let's kind of pull this back a little bit to the real basics. And when we start out with the real basics, we're talking about chromosomes. And so what chromosomes are the structures that store the genetic information in the form of DNA. And I'm old enough to have had traveling salespeople which would sell us encyclopedias. And I don't know if any, some of you probably remember that. Some of you probably don't remember that. And some of you may say, well, what's an encyclopedia? But chromosomes, I think of as being sort of the books that our genetic material is stored on. And so in this case, it's instead of having 26 different volumes of an encyclopedia from A to Z, you have 46 different chromosomes, 23 pairs of chromosomes. 22 of those pairs are what we call autosomes, and the same regardless of whether you're male or female. One pair are called sex chromosomes. Most phenotypic males have one X and one Y chromosome. Most phenotypic females have 2 X chromosomes. Again, there's some variability there. It can be complex, but that's what we see most of the time. And this is where all the information is stored. And so when we look at chromosomes under a microscope that have been stained in a certain way, this is what we call a karyotype. And this is what a normal female karyotype will look at. Because if you look at the bottom right corner, you'll see 2 X's. Most people, the vast majority of people with 2 X chromosomes are phenotypically female. And so how does this relate to DNA? Well, if we look at chromosomes, we find these are really very complex structures. And once you start unraveling those structures, you'll find these what we call coiled coils of DNA that are wrapped around these certain proteins. These proteins serve a functional and a structural role within that. But once you kind of unwind it far enough, what you will find is a single strand of DNA. And within those strands of DNA, there are genes. The genes themselves are individual instructions for that will cause the body to be able to do something, to make a protein that does something within the body. And RNA is the intermediate that will kind of function between the DNA part of this and the protein part of it. The RNA functions as the intermediary. Well, what is the DNA composed of? Well, the DNA itself is composed of what we call nucleotides. And you probably have heard the A's, C's, T's, and G's of the genetic code. Well, these are not metaphors. These are actually real structures. These are real chemicals that have a function within the body. And they are held together with a backbone that is made of phosphate and is also made of a sugar. If in DNA, it's a sugar called deoxyribose, and in RNA, it's a sugar called ribose, and those hold it together. And so the DNA is incredibly elegant in a lot of ways because the structure defines the function. The double helix is not just a cool way of visualizing DNA; it actually is the way it functions, and it functions in a way where that double helix is unzipped. And is unzipped from that DNA template, and then an enzyme. An enzyme is a protein that makes a chemical reaction happen, and that enzyme called RNA polymerase makes the RNA, and that RNA is kind of is formed on the template of that DNA, and you have to initiate it, and then you have to elongate it, and then you have to terminate it. So you have to start, you have to do the job, and then you have to stop it. And what you're left with at the end of transcription, which is an important step in all of this, is you're left with a molecule of mRNA. And you guys everybody here has probably heard a lot about mRNA because this is the technology used for 2 of the COVID vaccines. And mRNA is really important because you need it to go along and make the protein. And so I apologize for this sort of busy slide, but there's really only a few key points here. So you have your mRNA. It's like the messenger. Again, it's called messenger RNA that takes a message between parts of the cell. So the important parts for you to know is that the mRNA is read, and it's read by the body in these 3-letter words called codons. And each codon corresponds to an amino acid. But some amino acids have more than one codon. And so there's a little bit of tolerance there for certain changes, because there are 20 amino acids that you can make, and there are 64 different possibilities there. And what happens is the body reads the mRNA, and each mRNA through 3-letter words are read, and a new amino acid is made. And eventually, at the end of this, when the mRNA is totally read, you have a long strand of amino acids that go on to form a protein. And so this is a very basic way of just so you kind of helps you understand how changes to the DNA in a gene like ALPL can cause changes to the protein, that enzyme that can make it function in a different way, or in some cases make it not function. And so you can see there are 20 amino acids, and there are 64 different codons. And so in some cases, especially if that 3rd little letter of the word is changed, in many cases that's tolerated. And we call that the wobble base, and because that's a little more likely to cause problems. So how does this actually translate to how we practice genetic medicine? How do we actually work with patients in this and actually kind of describe what things do? And one of the ways we do this is with inheritance patterns. And there are two inheritance patterns that we can see with hypophosphatasia. One is called autosomal recessive. And so this is, in this case, this is the gene that's located on the autosome, not on an X chromosome. And we know that's the case for ALPL. So it's on chromosome 1. In this case, it means both copies of the gene are changed. They have a variant that's changing the function of the way the gene works. And in that case, you have a tendency for more severe disease. You have a tendency for this to be what we call more penetrant, meaning if you have both those changes on both genes, you are very likely to actually have the disorder. And both of your parents, in this case, would be carriers, meaning that they would have one of their genes that has the change. And I use the carrier in a very specific sense in that instance, because we also see autosomal dominant inheritance with HPP, meaning only one copy of the gene is affected. And this is really important in HPP. It's an important understanding is because we know from, or at least I'm going to back up a little bit. We think we know from research that's done so far is that about between 1 in 160 and 1 in 250 Americans have a change to one of their copies of ALPL. What we don't think is that all of those people have HPP. We think it's actually probably a small subset of those people that have one of those copies changed that actually exhibit signs and symptoms of HPP. So there are people with a change to their ALPL gene who do not have any symptoms. In my opinion, those people do not have HPP. If you have a change to your ALPL gene and you do have low alkaline phosphatase and you have signs and symptoms of HPP, then you do have HPP. Said another way is it is the problems, it is the symptoms of the condition that define the disorder, not just the genetic change. We have to be very careful about this because we want to make accurate diagnoses. It's always my goal as a clinician is to make accurate diagnoses of people with any condition. And we do see this sometimes, especially in dominant disease, where we'll see what we call incomplete penetrance, meaning not everybody that has a change will actually have the disease. So that's an important thing with HPP that's a little bit different. And again, when we're talking about homozygous and compound heterozygous, it can be very different. So homozygous means both copies of a gene are changed, and it's the same change. And so this is what was classically described in the Canadian Mennonite population. This one change on 2 copies. What we see more commonly with recessive disease here in North America, at least, is what we call compound heterozygous, meaning both copies are changed, but the copy on each gene, it's different. So it's not the same change on each copy. And then very commonly, what we'll also see is heterozygous copies. Again, one copy of the change is one copy of the gene is altered, the other copy is not. And so that's represented by the equal sign, or sometimes what will be called wild type. But I don't like that term, because it implies that there's a such thing as a wild type human, and there really isn't. But we use the term carrier and to describe the heterozygous state in people with HPP. I don't think the term carrier is really all that meaningful, except when we're talking about reproductive health in people with recessive disease. Then sometimes we have to use it to make sense of things. But I think for the person who's heterozygous for HPP, I think we have to be very careful about how we use that term, because we know these are people who may manifest disease. Again, not everybody does, but you can. So I think that's an important thing to keep in mind. And when we're actually looking at this, when we're actually doing the sequencing, we actually are reading through those bases. So we're reading through those bases, we're reading through those A's, C's, T's, and G's, and we're looking for a change about what is expected. And this has taken us a long time to get to. It's funny because it was actually not that long ago that we didn't understand how many chromosomes humans have. I mean, we're talking about the 1950s when we actually understood what the normal number of human chromosomes was. And it was until the 1970s before we could actually really read through in any meaningful way. And what's classically been done is to read through in a way that's very laborious. And what allowed us to be able to do what we do today is really benefits from the Human Genome Project. And the Human Genome Project started in 1990 and ended in 2003. Now, I'm too young to be, and I'm kind of the wrong kind of geneticist to be involved in the Human Genome Project in any way. But when I was an undergraduate, I was paying really close attention to this because I was doing genetic research at that point. And I would get, and this is an excerpt from an email I got in 1999 when I would have been a junior in college, where my research mentor said, I need you to be as specific as possible about the region of interest. We pay by the base pair, which, if you, from the 2021 perspective, is kind of crazy that we pay by the base pair, but we did. And for every 1 million base pairs, we paid $13,000 in 1999. The cost in 2021 per base pair is a lot less than that. And we'll kind of get to that a little bit later. But at that point in 1999, we were using what was called Sanger sequencing. This is that technique that was developed in the late 1970s, and it's very good, but it's very slow. And many of you who had testing done before probably about 2010 to maybe even 2012 almost certainly had your genetic testing done with this technique called Sanger sequencing. Again, Sanger sequencing. It's very it's good. It's good at identifying the change. If you're looking at one gene of interest, it's very accurate. It's slow. It's time consuming. It's laborious, and so it is. So it's a little bit limited. So I mentioned to you that in 1999 the cost of sequencing 1 million base pairs of DNA was $13,000, and we saw the cost decline kind of in a linear fashion. And the linear fashion resembled very closely what was called a different rule called Moore's Law. And Moore's Law really didn't have anything to do with genetics. It had everything to do with computer technology. And so a number of years ago, it was observed that computers both doubled in power and decreased in price about every year. And then eventually that was every 2 years, which held true for a while. And genetic sequencing technology really mirrored that until about 2008. And since then, it's way exceeded that. And what this is the reason why this happened is what we call next generation sequencing. And this is really what we use at this point instead of just Sanger sequencing. But what we've eventually had to do as a part of the Human Genome Project is go from automated Sanger sequencing, which helps a little bit, to kind of early parts of next generation sequencing to a much more mature platform of next generation sequencing. So basically what next generation sequencing does is, instead of taking one strand of DNA and being very laborious about sequencing that and knowing exactly what you're dealing with, you're sequencing lots of different strands of DNA simultaneously, and then you have a computer basically put it together and try to piece out the map. And some of you may have heard this described as shotgun sequencing in the late 1990s and early 2000s. That's kind of why they call it shotgun sequencing, is you're sequencing everything at the same time and have the computer sort of clean it up and see what things look like. And that's actually pretty helpful. And by 2014, what it resulted in was a $1,000 genome. And the $1,000 genome is fraught, because that's $1,000 genome means you can produce a sequence in $1,000. Doesn't mean your particular genome that you get from a genetic testing company costs $1,000, because there's a lot more caveats there. But our stopgap has been exome sequencing. And so exome sequencing is, instead of looking at every gene, every piece of DNA, you're just looking at the parts of the DNA that actually contain the genes. That's about 1.5% or 2% of the total genome. And so you actually will get some benefits from that. And that's gotten a lot cheaper too. And so it used to be that this was very, very expensive. And now you can actually get a raw sequence for a lot cheaper than this. But exome sequencing is a lot of times used if we don't know exactly what's going on. We suspect something's genetic. We don't know exactly for sure. But it helps us, so we're able to do a lot of different genes simultaneously. So if you don't understand what's going on, or if you don't really have one gene of interest, it might be a reasonable thing to do a whole exome. But the problem is it's not really whole exome. It's probably only about 90%. And we think that there are still areas where there are no genes that are still really important to disease. And so it's still not getting at all the things that we want. And there's still a lot we don't understand about the genome. There are about probably 21,000 human genes, and about 4,000 of those genes have a disease associated with them. So there's a lot we still have left to know. And kind of a fun fact, we actually still don't know exactly how many genes humans have. And this is, we are now 18 years after the conclusion of the Human Genome Project, and we still don't agree on it. It's funny because before the Human Genome Project, the estimates were that humans had between 80,000 and 100,000 genes. We're humans. We must have more genes than these other animals. As it turns out, it's not true. The range is narrowed somewhere between 19,000 and 22,000. So you'll hear me say things like 20,000 or 21,000 to kind of split that difference in what we think we know. So just to give you an idea, how much does whole genome sequencing cost? Minus interpretation plus caveats. The first sequence was $2.7 billion as a part of the Human Genome Project. As of this month, if you're, I can get you a whole, at least the raw data from a whole genome for $300. So it's come down in price just a little bit. What about HPP in particular? Well, we know that HPP is caused by changes to the Alpl gene. Alpl gene is actually a pretty small gene. It's not very complex. And there's only 12 exons. So exons are the little chunks of the gene, and only 11 of those are actually in the protein product. And there's a lot of different changes that we've seen. And really what we're looking at is spelling errors in most cases. This is probably a little over 80% of the changes in ALPL are little spelling errors that we actually will have to read through the gene. And that you can read through the gene with Sanger sequencing, you can do it with next generation sequencing. But about 17% of the time, we're actually looking for larger chunks that are missing or extra. And this is important to do in a lot of cases. But in my opinion, somewhere between about 5% and maybe even as high as 15 or 20% of people who we really think have HPP don't have any changes identified. And I think that's very interesting to look. It shows you that we're still learning about HPP and we're still learning about how this gene works. Now, this is a crazy matrix here, and I'm not going to go through this. All I want you to do is just appreciate that there's a lot of different moving parts here when it comes to interpreting genetic testing. And so many of you have had genetic testing and will have variants identified that will say things like pathogenic or likely pathogenic, and we view those as being disease causing. But a lot of you may have had something that we call variant of uncertain significance. And the reason why you have that is our guidelines for genetic testing interpretation are really, really conservative. We'd rather give you ambiguity than give you the wrong answer. And that may be maddening, but I still think in most cases it's the right thing to do, because we don't want to have to say, well, we think this actually probably is really a change, and then have to backpedal. That's no good. And just so everybody knows what a genetic testing report looks like. This is a good report. And it tells you a few important things. So if you're going from the top, it tells you where you got the DNA. In this case, it's from blood. It tells you what test was done. So this is our kind of flavor of whole exome sequencing done at Children's Mercy. And it tells you that what state it's in. So in this case, heterozygous. It tells you what the interpretation of the change is. In this case, it's pathogenic. And it tells you the gene and the exact change. It tells you what the DNA change is, and it tells you what the protein change is. It also says no second variant was identified. So say that this individual at least a carrier. So at least a carrier implies that there could be another change there, but it's also possible that a person who was heterozygous could also have disease. And it tells us what else was looked at. So that's important. But whenever we're looking at these things, we also see variants of unknown significance. So when I look at variants of uncertain significance, they used to really bug me, and I used to break out into a cold sweat. But I mean, we've been dealing with this for so long that it doesn't really bother me anymore. But we're looking at the individual gene, and it's important for us to know what kind of stuff is going on with this gene. What does this gene actually do in the body? What happens when this gene goes wrong? And does that seem to fit the patient? Because that's really important to know if this is something that fits the patient. And you'll see these things that are, again, if you look at this LRP5 change, and you'll go down to the first subpoint here, what you get is a little bit of a kind of a window into what we think might be going on. So we use some tools that are computer-based tools that'll predict what they think is going on. In this case, they say, well, we think this change is probably going to be okay. It's either tolerated or benign. We see the highest frequency, and these are our big databases looking at lots of different humans to kind of build an idea of what the normal variation for humans look like. And so we see, well, a couple people have this change. And so we look at this and say, well, that's not super suspicious. And so a lot of times we'll say the word I use is bland. It's a bland variant. And that can be really helpful to kind of know that this is something that maybe you're not super suspicious about. Whereas sometimes you'll look at a variant and you will say, gosh, that looks really, really not good. That's maybe the parents didn't have it, or maybe it's predicted to be pathogenic, or it's really rare. Those are things that we might be very concerned about. So there's a real art to interpreting those variants of uncertain significance. But I think it is important to know that this is kind of just a fact of life when we're doing this sort of genetic testing. And so in conclusion, so we can have a few minutes for questions, my opinion is I think we've learned a lot about HPP over the past few years. I think we have a lot still to learn, but I think we've learned a lot. And the other thing I would say is I feel like my understanding continues to be as a multi-systemic inborn error of metabolism, not just a bone disorder. And I also think it's helpful to understand a little bit about the genetics and genomics when we're interpreting genetic testing. And hopefully this helps kind of give you a window into not only about the genetics and genomics, but just how we think about genetic testing for our patients with HPP. So thanks very much. I appreciate the opportunity to talk to you this morning. I wish we were in person. I'm certainly looking forward to next year in Orlando.
Yeah, thank you so much. That was so educational. I learned a lot. So I appreciate you going into some of that detail. I think a lot of us who have been to these talks before learn something every time we hear you speak. So thank you so much. We do have some questions. One of them actually came from the Facebook page. Discussion happened over the past week. A patient posted, I have two mutations. I just found out I have a 50% chance of any future child resulting in having HPP. Basically, I've been told all my life I inherited it through autorecessive, but can one parent have a recessive gene and the other a dominant? I'm just trying to understand where the 50% came from.
Yeah, that's a really good question and a really important one. There's a little bit to unpack here. We in genetics very seldom say things like 0% or 100%. But if a person has recessive HPP, they only have those copies that have a change to them. So you will pass on a copy that has a change to them. It's a 50% for each one of the changes. Mm-hmm. But so if a person has recessive disease, there's a 100% chance that you're going to pass on one of those changes to your kids. Mm-hmm. Now, they may not have disease. They may not have disease. What's important to know is if that person's partner has a change to one of their copies, if they're an asymptomatic, what we would typically call carrier, mm-hmm. then we get into a very different set of discussions. But so if they're not a carrier, then we know it's 100% chance that the offspring of a person with recessive disease is going to have one of those copies. And then it's kind of what is the risk of a person with heterozygous, who's heterozygous for one of those changes is going to have disease? We know it's not 100%. And so I would say it's 100% chance of them inheriting that change. It's not at all 100% chance that they're going to have disease. Okay. As far as we can tell, it's probably a relatively low penetrance, maybe even single digits %. We don't exactly know. But this is one of those cases where you'd say it's 100% chance that you're going to pass on one of those copies.
Okay, great. There's a couple questions that hover around the same topic, so I'm going to try to combine them. Patients who have a single mutation are adults. Mm-hmm. May have pain, may not have bone manifestations of HPP, but they're looking at that, they have a low ALP, looking at that question of what's the right time to start therapy. Mm-hmm. I think that there's some confusion sometimes because, you think if the enzyme's low, you would want to take an enzyme replacement therapy, but that's not always the case with HPP. So maybe you could just talk a little bit about what goes into weighing the whether or not a patient should be a candidate for therapy.
Yeah. So I guess my comment would be I treat patients, not lab values. Mm-hmm. And so I look at this much more from the standpoint of what is actually happening to the patient. Are you having signs and symptoms? And so I kind of base the therapy, whatever that therapy is, whether that's a medical therapy, whether that's a rehabilitative therapy, I really base that on what the patient is experiencing. And I don't look at it as much as do you have low low ALP, because in all honesty, if you're totally asymptomatic and just have low ALP, I don't know what to follow. I don't know how to follow you clinically. I don't know how to look for improvement. That concerns me. So I would say, the individual experience is going to be varied, and so I can't necessarily speak to any one person's individual experience, but I would just say kind of that's my personal paradigm of treatment.
Yeah, we've told a lot of patients maybe, just even to get a baseline. So as you continue to age and maybe, as women go through menopause, you have something to at least see if you have any kind of progression of disease, which to your point would give you something to look at.
Mm-hmm.
Yeah.
Yeah.
Okay. So there's one more question, and this is an interesting question about whether or not there's such a thing as HPP, flares or any kind of symptoms or fractures. Have you seen any evidence that there's flares that go dormant for a time in childhood or adult and then return? This has been my experience, and just wondering if you have any theories about what could be the cause of that.
I have noticed that in many cases, patients who have had symptoms as children will have a period of time, often during sort of adolescence into young adulthood, where they are relatively asymptomatic. This may have to do with the expression of ALP that happens. Everybody has different ALP levels at different ages, and during kind of pubertal development, the ALP is relatively higher in everybody. So you might have my ALP right now may be about 70. Whereas when when I was 15, I know it was 400. And that's true for everybody, that there's kind of a curve over time, but it's different for people with HPP. It may not be as high as what you might experience, but it may be enough that you may be getting some added bone mineralization. You may be getting some other activity, and so it might be enough that the symptoms will be a little quiescent over the next few years. So it wouldn't be a rare thing for me to observe a person who kind of starts having some improvement in their symptoms spontaneously in adolescence, and then it seems like anywhere between sort of late 20s to mid 40s that we can start seeing kind of reemergence. It's not universal. Not everybody's experience is different, but that's definitely some observations I've observed.
Great. All right. Well, that's it for the questions right now. And for those of you who have attended, you have to actually leave this session and go back to the agenda and click on the next segment. So, Dr. Rush, thank you so much, and we'll see you in the next session.
Thank you. Appreciate it. See you in the next session.